Hello, BIO178. This video is going to give some background about the dataset that we're working with for our single population genetic diversity lab. And this is actually a dataset that's very near and dear to my own heart because it was a dataset from my own dissertation work. Okay. So the study organism that we're working with that I've talked about a little bit already in some of the lecture videos is the intertidal snail Chlorostoma funnebralis, shown here. Definitely not to scale. They're small. They can fit in the palm of your hand. And the sites that I've been working at in the studies I've been doing the past 8-ish or so years is specifically looking at different populations within California. And we'll talk later in the semester how I'm really interested about some of the environmental differences at these sites, but for this lab we're focusing on, just like we talked about in lecture, some different parameters of the different populations. So we'll focus on the populations themselves as entities. And specifically, the data set that we're working with today was collected from 6 different geographic sites in California, 3 in Northern California that I'm showing here in blue. So Slide Ranch, Pescadero, and Pigeon Point. These are all within driving distance of Sacramento. There are the abbreviations that you'll see if you look at the data file. That I'll get to that point later. I'll explain more, but these are the abbreviations that you'll see for each of those sites. So SR is Slide Ranch, PES is Pescadero, and PP is Pigeon Point. If you're ever looking for something to do, Slide Ranch is a great site. It's one of my favorite sites. It's actually on a farm, like an educational farm, but you can walk down to the water and they have really great tide pools. So those are the 3 Northern California sites. And then I also have 3 Southern California sites. Aliso Beach, which is in Laguna Beach, right next to it, La Jolla, and Bird Rock, which are both in the San Diego area. And the abbreviations for those are AB for Aliso Beach, LJ for La Jolla, and BR for Bird Rock. All those Southern California sites are going to be in red. And then for the data set in particular we're working with today, there are 15 individuals from each of these 3 northern sites and 15 individuals from each of these 3 southern sites. And the data set that we're working with is obtained from a ddRAD high throughput sequencing run. So we're working with high throughput sequencing data again in this lab. Remember, or if we haven't talked about it yet, RAD-seq is a type of high throughput whole genome sequencing, but it's a type of reduced representation sequencing, which means you're looking at markers spread across the entire genome, but you're not sequencing the entire genome itself. That's mostly for cost purposes. If I could have sequenced the entire genome for all of these guys, I would have. What does ddRAD stand for? It stands for Double Digest Restriction Site Associated DNA. Again, that's because the method that's used to cut up the genome into smaller pieces is restriction enzymes. So that's what's shown here. That's where that name comes for this particular sequencing technique. Restriction enzymes are used in preparation of the samples before you actually do the high throughput sequencing. Once you do that you get the reads back, there's a program, this has already been done, but the program, what it does is reassembles the short reads that the sequencer gives you. We'll cover this in more detail later in the semester, but the main thing that we're focusing on in terms of molecular markers in this data set is SNPs. So what the program does is for each different locus, and you'll see in the data file, these are given these number designations as what the program gave them. So you might see a number like 5750, that just refers to one particular locus or one particular SNP. And what the program does is line up all of the DNA sequences from all of the individuals from Northern California, all the individuals from Southern California, and then identify SNPs or single nucleotide polymorphism differences between those 2 sites. So here highlighted in yellow, I have one site where all the Northern California populations have a T nucleotide, and all but one of the Southern California population individuals actually have a different nucleotide, a C. So this is a SNP that's been identified in the data set. This particular SNP would be designated as 5750, and then the program does this for the entire ddRAD sequencing set. And so for my particular data, I ended up with about 1,800 SNPs. We're just working with a subset of that today. When I was working through this lab yesterday, I accidentally asked R to analyze all 1,800 SNPs, which it can do. It'll just take a really long time. So for our purposes, probably not great for a time-constrained lab. So what I did is pull out a subset of that data for us to work with. Let me move my head so you can see the data file. So this is a screenshot here of the data file I'm going to be giving you to work with. It's in a file format called GenePop, just FYI. What this subset data contains is 8 different SNP loci, and we have information for all 90 individuals. Remember, we had 6 of those geographic sites and 15 individuals at each site. So if we look at this data file here on the right, those are the different loci that I talked about. So we have 8 different SNP loci. That's what's indicated in each of these 8 columns here. Each row is a different individual, and here are those abbreviations I talked about. So PES, PP, SR. And what we do in this file is we designate with this pop header where we want to break up these different populations. I'll talk in a couple slides about why we're choosing to break it up in this particular way. What we're doing is essentially treating this data set as 2 different populations, a Northern California population and a Southern California population. So each row is an individual broken up into the 2 different populations we're comparing. Each column is one of those 8 different SNP loci, and then the numbers here just indicate the particular genotype for that SNP. If there's a 0, that means we actually didn't have data for that particular individual at that particular SNP site. And then the numbers here correspond to different base pairs. Remember, we have A, C, T, and G. That's why they're up to 4 numbers. If you have the same number there, that means that both alleles for that individual had the same nucleotide. Different numbers mean it's a heterozygote. So that isn't super critical for the actual lab we're going to be doing today, but just so you have a basic understanding of what the actual data is that we're working with. And let me move my head again. So like I said, in this data set, we're comparing, or in this lab, we're treating those 6 geographic sites as 2 different populations. And we've talked about this a little bit in lecture this week, where we said that oftentimes genetic data can help us inform how many populations we actually have. This is some SNP data I've shown a couple times now. And in these trees, each tip is a different individual, and it's color-coded according to whether they're from Southern California in red or Northern California in blue. This tree on the left is the one constructed from all 1,800 SNPs in the data set. This one is constructed just from the outlier adaptive SNPs that we found. And in either case, we're not seeing any separation by population. So if you look at these abbreviations, so BR, PES, for instance, if we had 6 different very discrete populations, what we would see is whole clusterings here that only contain individuals from one of those geographic sites. So we would see clusterings with all BR individuals, for instance. And even when we're only looking at the adaptive outlier SNPs on the right, even when we do start to see some clustering, it's only between Northern and Southern California, and it's still not within these 6 individual geographic sites. So that's why in this data set, we're treating those 3 Northern California sites as one population, and then we're treating those 3 Southern California geographic sites as one population. So we're comparing 2 different data sets in this particular lab.